Good day. My name is Osita Dema Ubok. I'm introducing you to principles of biotechnology. Biotechnology is a field. If you look at the world, you will see that it was derived from two keywords, bios and the technology, biology and technology. Bios means study of life. Technology itself is derived from two keywords, technos, meaning to create, and logos, meaning study of. The UN Convention on Biological Diversity in 1992 defined biotechnology as any technological application that uses biological systems, living organisms, or derivatives thereof to make or modify products or processes for specific use. Biotechnology is concerned with applications of biological systems, living organisms, their derivatives, using technology to make or modify products or processes for specific use. It is an exciting field today that borrows from various disciplines of microbiology, biochemistry, molecular biology, anatomy, physiology, food science, chemical engineering, and so on and so forth. In 1981, the European Federation of Biotechnology defined biotechnology as the integrated use of biochemistry, microbiology, and chemical engineering in order to achieve technological applications of the capacities of microbes and cultured tissue cells. Biotechnology involves cell and tissue culture techniques, enzyme technology, and genetic engineering. There are various definitions. The United States National Science Foundation defined biotechnology as the controlled use of biological agents such as microorganisms or cellular components for beneficial use. There is a definition by J.D. Bullock, and it defined biotechnology as the controlled and deliberate application of simple biological agents, whether living or dead, either cells or cell components, in technically useful operations, either of productive manufacture or as service operation. Gibbs and Greenhill, in their own definition, stated that biotechnology is the use of living organisms in systems or processes for the manufacture of useful products. Generally, biotechnology is a combination of scientific and engineering principles, utilizing biological agents to process materials into value-added products or commercially viable products. If we look at all the definitions stated, we will observe an emphasis on the following: the use of living organisms or their products in one way or the other, the manipulation of living systems, which means there could be a change from what is normal or natural. And then the essence is always aimed at towards producing something that is beneficial to humans. Biotechnology is broad range and it's an interdisciplinary activity. Interdisciplinary subject encompassing all the disciplines of basic sciences and engineering and draws heavily from microbiology, biochemistry, molecular biology, immunology, cell and tissue culture, physiology, etc. It leans heavily on chemical and biochemical engineering because mostly large-scale cultivation of microorganisms and their cells or cells and tissue cultures and the harvesting of the products involves downstream processing. Therefore, a good understanding of the basic principles of chemical and biochemical engineering is important. Generally, biotechnology can be divided into traditional and modern biotechnology. Traditional biotechnology is usually used to refer to the traditional techniques that involve use of living organisms to yield products or modify foods or other useful products for human use. The early examples of biotechnology include making of cheese and yogurt, making of beer, wine, bread. There are also examples of breeding of animals and crops. Some traditional techniques, such as hybridization and the mutagenesis. The diversity of microorganisms and development of genetics expanded the potential of traditional biotechnology. And this had led to development of what is referred to today as modern biotechnology. Modern biotechnology involves the use of genetic engineering techniques such as recombinant DNA technology, functional and structural genomics, genetic modifications for the development of products and processes. Modern biotechnology has contributed significantly to enhance our knowledge of biological systems. Genes have been sequenced and the specific genes can be identified and these genes can be modified and specific and desired products can be made through the processes of modern biotechnology. One of the most extensive applications of modern biotechnology is in the field of agriculture and medicine. Recombinant DNA techniques and mutagenesis are used in agriculture to develop plants with novel traits. New drugs, especially targeted therapy, can also be achieved using modern biotechnology. Generally, biotechnology can also be broadly divided into cell and tissue culture techniques, enzyme technology, and genetic engineering. There is a thin line separating these broad classifications. Because for you to harvest enzymes, you have to most times begin with the cell and the tissues that produce the enzyme. And for you to manipulate genes, you have to identify the organism, the cells that harbor the genes, and identify the specific gene, isolate it, and begin to improve the gene. Let us briefly look at cell and tissue culture. Cell and tissue culture is the base of biotechnology. Primarily, fermentations were usually based on cells that carried out fermentation. However, a cell culture is basically keeping the cells taken out of a source in an artificial environment alive. The cell cultures are grown in vitro, and the cells can be used for production of monoclonal antibodies, vaccines, enzymes, hormones, interleukins, and growth factors. Cell cultures are very important in the development of new drugs, even in cell or stem cell therapy methods and toxicological studies. Tissues can be cultured. Tissues are just fragments of an animal or plant and can be grown in artificial environments in which they can continue to survive and function. And the cultured tissue may consist of a single cell, a population of cells, or a whole part of an organ. And cells like meristem cells, plantlets are important in biotechnology. Enzyme technology broadly involves production, isolation, purification, and use of enzymes, either in soluble or immobilized forms, for the ultimate benefit of mankind. Various enzymes are found in nature, and since ancient times, enzymes have been applied knowingly or unknowingly in food production, production of cheese, sourdough, beer, wine, vinegar, and even in the tanning of leather. Most of these processes mentioned relied on either enzyme produced by spontaneously growing microorganisms or enzymes present in added preparations such as papaya fruit or even cow's rumen. The development of fermentation processes with selected strains of microorganisms is dependent on the enzymes produced by those organisms, and they are used to carry out fermentation. The enzymes do bioconversion for production of the desired products. Genetic engineering is also called genetic modification. It's a process that uses laboratory-based technologies to alter the DNA makeup of an organism. And it may involve just changing a single base pair or deleting a region of DNA or adding an entirely new segment of DNA. Various processes are involved, DNA extraction, gene cloning, and then gene modifications. And all these are the basic aspects of biotechnology. Because of the rapid growing nature of biotechnology, it has an its application in various fields of human endeavor. Biotechnology is therefore classified into various types. The health and medical applications of biotechnology is referred to as red biotechnology, as exemplified by gene therapy, drug development, and etc. The application of biotechnology in human welfare and leisure is referred to pink biotechnology. The typical examples are anti-hangover probiotics and the glowing bacterial lamp. Generally, the application of biotechnology in agriculture and farming is referred to as green biotechnology. Biotechnology is used to develop pest-resistant crops, probiotics for farm animals, biofertilizers, crops that are resistant to drought, etc. White biotechnology refers to industrial and manufacturing applications of biotechnology. Biotechnology can be applied in production of biopolymers, production of biofuels from microalgae. The environmental applications of biotechnology is referred to as gray biotechnology, exemplified by bioremediation of chemicals and gene-derived control of invasive species. Yellow biotechnology is used to describe food processing and nutrition applications. Processes like brewing, production of lactose-free dairy. Brown biotechnology describes applications aimed at improving living conditions in arid and desertic areas. Development of drug-resistant crops drought-resistant crops, I beg your pardon, is a typical example. Blue biotechnology is application of biotechnology as sustaining water resources. And examples include the development of genetically modified fishes for fish farms, wastewater treatment processes. Of course, some persons refer to it as marine biotechnology. The applications of biotechnology aim to improve the processing of biological data is referred to as good biotechnology. Examples are software and model development, standardization of laboratory practices and protocols. Purple biotechnology is used to refer to applications aimed to connect biotechnology with the rest of society. Ethics, intellectual rights protection, issues of laws and regulations. The application of biotechnology in bioterrorism and warfare is referred to as dark biotechnology. The example is the development of biological weapons, pathogen release, and light biotechnology is the aspect of biotechnology that is used to protect against accidents and misuse of biotechnology. Development biocontainment systems and biomonitoring. Biotechnology has rapidly emerged as an area of activity that have enormous potentials for human welfare, ranging from food processing in medicine, in agriculture, animal health and husbandry in medicine, production of chemicals, even crime detection and parentage disputes, renewable energy, production of biofuels, applications in biomining, in fisheries and aquaculture. In fact, typical examples of contributions of biotechnology to human welfare include the production of monoclonal antibodies used for diagnosis, e.g., diagnosis of hepatitis B and other viral diseases. DNO probes are also used for disease diagnosis. We also have recombinant DNA vaccines, which are said to be cleaner, safer, and production of valuable drugs like human insulin, human interferon, and bovine growth hormones. Today, gene therapy is touted to cure that it can cure several diseases and has been applied in Huntington's chorea, cystic fibrosis, and artificial insemination is used to select sex of babies. In the industrial system, useful products, improvement in production of products like ethanol, lactic acid, glycerin, citric acid, gluconic acid, antibiotics, enzymes like alpha amylases, proteases, lipases, production of single cell protein from yeasts for human and animal feeds are very important. Management of oil spills, detoxification of waste and industrial effluents, using techniques of bioremediation. Then in the animal biotechnology, in vitro fertilization, of transgenic animals for increased milk production and the meat production is notable. Today, biotechnology is so important that we are in the era of gene revolution, the era of molecular biology, where you can specifically identify the gene that does what and can use that gene. Either you amplify it or the gene can be deleted or it can be manipulated to improve uh, the welfare of humans or animals. But ultimately, whether you are improving agriculture, in medicine, food production is for ultimate benefit of mankind. Thank you, and ensure you listen to the next lecture.